Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. As always, we have with us today Dr. Rajesh or Raj Devraj, who is the CEO of Rectify Pharma, which is a company focused on advancing positive functional modulators, or PFMs, which is a novel class of oral small molecules that restore and enhance membrane protein function linked to various diseases, including those of the liver, cardio-renal-metabolic, and neurodegenerative diseases. In addition to serving as the CEO of Rectify Pharma, Dr. Devraj is a venture partner at Atlas Venture. Dr. Devraj has over 30 years of experience in both large pharma and biotech, including leadership roles at Pfizer and Deciphera, and he's also built a number of biotech companies with Atlas, including Padlock Therapeutics, and he also co-founded Disarm Therapeutics with Venture, where he served as its chief scientific officer prior to its acquisition by Eli Lilly. Dr. Devraj is well known for his drug hunting capabilities and deep knowledge of drug discovery, early clinical development, and strategic planning, teams that have helped advance multiple candidates into clinical trials for diseases including refractory cancers, autoimmune diseases, COPD, and pain. Dr. Devraj holds a Bachelor of Science in Pharmacy from the University of Mumbai and a PhD in Medicinal Chemistry from Duquesne University. To hear more about the innovative work that Rectify Pharma is advancing in the liver disease landscape, let's turn to Dr. Devraj now. Dr. Devraj, thank you so much for being on the show today.
No, thank you. Thanks a lot. Thanks for giving the opportunity to talk about Rectify.
Thank you for making the time. Dr. Devraj, having spent a number of decades in the field, I think we really should talk about how you've built up to becoming the CEO of Rectify Pharma. And as I mentioned, that you've been involved in a number of multiple successful biotech startups, and you have extensive work in the drug development field. So what have been some of the key experiences and learnings from those decades of work that you've brought to Rectify Pharma?
Yeah, I think good question. A number. Let's just hit on maybe two or three key ones. I think number one is the ability to synthesize a vast array of information and knowledge from various disciplines and bringing them to bear in terms of really understanding, is there a drug product to be had? Number one. How do you build a drug product that can meet a target product profile? And does it really bring value to patients? So really sort of marrying the early parts of drug discovery innovation and through to an early clinical planning and development strategy where you can really test the compound very quickly in patients is something that I bring to the table. And of course, then it's all about really putting the plan together, understanding what teams you need to be building. Number two, what sort of finances you need that you need to get for the company. And number three is what sort of corporate strategic objectives you have. What are the things that you want to partner? What are the things that you want to keep and take forward? And of course, that depends on the climate that every biotech company is living in, not only internally, but also in the external world.
And so what would you say the climate is currently in the biotech space, and how has it evolved in your time in the field, and where do you see it heading?
Right. 30 years in the business, right? So equally weighted, 15 years in large pharma and 15 years in biotech. So I guess I have but the last 15 years has been the recent experience is all about biotech. And I've gone through the ups and downs of different cycles of biotech building as well. And I can tell you at this current time, it's no new news to anyone, but the climate is hard, right? But it is a a dichotomy, as we all heard in the JPM of this year, where there seems to be still a good amount of capital that flows into strong science held private companies. It is not the same for public companies, as you can all imagine, because there's so much other places to find and put your capital to work in terms of generalist investors. So, where I stand in terms of Rectify Pharma, which is a private company, we are very well capitalized to take us to a clinical milestone. So, just very different, I would say, in terms of capital environment. Um, and also, I would say investors are looking more and more for asset based companies versus building very large platform companies. And again, it's no new news to anyone, but as you can all tell, sort of the modality, loving new modalities has come in ups and downs or different cycles. It takes a very long time to innovate. And as you all know, platform and gene therapy companies and finding new gene therapy modalities has not been very easy in terms of building companies and raising capital for those. But it still is required. I think there's tremendous room to innovate there, and think you definitely need a lot of capital, a lot of talent, and good science to break through to help us innovate new medicines for patients.
Right. And so, really pioneering new medicines and new avenues and approaches for patients. Can you tell us a bit about Rectify Pharma's mission and goals, and really its approach to the space that it's working in, including liver diseases?
Right. So let's start at the front, right? The story begins with trying to find n- new modalities, which are small molecules that can restore or enhance membrane protein function. So you start with that premise. And one of the key foundational innovators in this space has been Vertex. Vertex has developed what are correctors and potentiators for cystic fibrosis and has really built a very successful franchise and helped deliver drugs that have really uh, provided a profound impact to cystic fibrosis patients. So we sort of stood on the shoulder of giants. We have a precedented modality in small molecules that Vertex had built. What we wanted to do was to take a more general approach and understand whether we can use a similar approach for multiple different membrane proteins. We started out initially with ABC transporters because CFTR gene is nothing more than ABCC7. So again, building on the precedence and understanding, is there room for us to really drug ABC transporters and understand whether we could restore or enhance protein function of ABC transporters. So we started with that space. So we wanted to build out a platform that was general and that could find very efficiently and in a very in a rapid manner, small molecules, which we term as positive functional modulators that can restore back or enhance protein function of ABC transporters. And then we started to understand both from a top down and a bottoms up approach, what are the top targets within ABC transporter family that we wanted to work on. There are 48 ABC transporters in the human proteome. 21 of the 48 have etiologic mutations that cause disease. So strong human genetic evidence because these cause rare monogenic disease. We began there because were building a new platform. We wanted to de-risk sort of the biological target thesis, and we started working on a few targets there. As we started to build a platform and started to screen our small molecule library that's ever growing and, of course, increasing in knowledge, we found out that our small molecule positive functional modulators can not only correct the mutations, but is also able to enhance wild type function. That quickly opened the door of the therapeutic opportunities. We could walk from rare monogenic to haploinsufficient genetic diseases all the way to large, rare, large common diseases. So you could sort of go through the spectrum with our small molecule PFMs. So what we have currently is a lead program, which is a dual targeted positive functional modulator that targets two ABCB family transporters, BSEP or ABCB11 and two ABCB4. Both of these are expressed in the liver and play a very important role in maintaining bile acid homeostasis and bile acid composition in the liver and the biliary tract. So, what it became very evident to us is that this dual targeted PFM has the potential to be a pipeline in a pill for multiple hepatobiliary diseases. We chose PSC as our lead indication, primary sclerosing cholangitis. It's a large, rare, unmet disease. It's predominantly a bile duct disease that causes cholestasis as a secondary event. So it's basically it's the strictures of the bile duct that leads to bile acid toxicity, bile acid homeostasis leads to fibrosis, eventually leading to, of course, cholestasis, liver cirrhosis, and liver failure. We have a very unique approach to treating PSC because of the dual targeted mechanism. What BSEP does for a living, it effluxes bile acid away from hepatocytes into the bile duct. So we are positively increasing the efflux of bile acids, so removing the toxic bile acid accumulation within the liver and helping sort of with the improving the bile flow away from the liver into the biliary tract. But that is necessary but not sufficient. What we also bring to the table is a very, very important and I think powerful mechanism, which is B4 modulation. What does ABCB4 normally do? It effluxes phospholipids. And why is this important? Phospholipids are very important to buffer bile acids and buffer free cholesterol. As you know, free cholesterol is a potent anti-inflammatory molecule, whether it's in the circulation or in our case in the biliary tract. And also, as you know, free bile acids are very detergent to the bile tract, and they cause a lot of epithelial toxicity to the bile epithelium and biliary tract. So by modulating B4, we are able to change the composition of the bile to more normal bile. So PSC is known as having a functional deficit in phospholipids. So we are directly addressing that as well with B4. So this combination of this mechanism of BSEP modulation and B4 modulation allows us in one molecule to address abnormal bile acid composition and also address accumulation of toxic bile acids within the liver and improving bile flow. So we think we have a very powerful mechanism that can restore back normal biliary health and also prevent that way cholangitis and cholestasis that sort of underlies the pathology of PSC.
Wow. Very kind of elegant, as they say, approach, and to really first targeting the underlying mechanism of disease. And that's exactly what, when we talk about targeted therapies and personalized medicine, really that this really speaks to that. So these PFMs, and you mentioned that right now, your lead candidates are focused on targeting ABC transporters, but could this also be targeted to other proteins and to enhance, of course, other protein functions? And what other kind of proteins would you be looking at similarly?
Right, correct. So let me sort of play out two or three other programs that we've worked on. Our second program is also an ABC transporter targeted therapy for large cardio-renal indications, including CKD and vascular calcification that underlies much of the morbidity and mortality that you see in ESRD patients. So we are developing a therapeutic, which is an ABC6 positive functional modulator for both CKD and preventing the renal insufficiency that comes in CKD patients. Again, a very targeted therapy. We know exactly the target role and why that's going to be linked from target to mechanism to effect in protecting kidney function. So C6, and then we have an ABCD1, D2 targeted PFM for treating XALD and AMN, a very large rare neuro opportunity, about 20 to 25,000 patients. Beyond ABC transporters, over the past 9 months or so, we have been opening up the opportunity of exploiting the platform for other membrane proteins. And these include A, ion channels, and we're going to talk a few about that. Number two, solute like carrier proteins. Number three, GPCRs. And number four, ATPases. All these proteins are also using trafficking as a core mechanism for getting from ER to the Golgi to the cell membrane where they can function properly. So we are basically exploiting the same mechanisms that we've used for ABC transporters to really start to address some of the important diseases that are mediated by protein dysfunction of these other classes of proteins that I discussed. The most predominant among them is ADPKD. Right? Autosomal dominant polycystic kidney disease, which is really driven by two proteins, PC1 and PC2, both of which are ion channels and play a very important role in maintaining cyst health in the kidney. So I think we have a great, we've already started to develop some ideas, and we have some early small molecules that are moving forward for ADPKD as well. Similarly, we have other programs that we are targeting at this point in time with other transporters.
Wonderful. That's one great overview. Thank you so much for that. And really, again, like being able to translate and to take this concept of really enhancing protein function. I think it's very novel, and it's very it definitely makes sense to do so and to really target the underlying causes of the diseases. So really fantastic to learn about the biology and the intricacy of it all. And so I'd like to maybe take a step back and ask you about really what are the current challenges in early drug development. I think we hear a lot about the challenges going on into clinical stage and clinical trials, but I feel like really in that early phase is where you have to really hone in on how you're going to move forward with your programs. And you being in the field and being dubbed a drug hunter, I think it was, I was reading. And so how do you identify those compounds, and how do you hunt them down, and how do you really decide which ones to move forward with, and how do you face the challenges in sticking to the drugs that you think will make it through?
Yeah, I think you asked a big question. I wish it was a
It's like, tell me
simple
about your
answer.
30 years of drug hunting in 2 minutes.
I know. It's unfortunately it's a different solution for every different
Hmm.
target, and especially with every different modality. And small molecules have their own set of challenges and also opportunities, right? Strengths that they offer. Um, let's maybe just hone back a little bit. I think one of the key things that I've worked with, it's number one, people, top notch people that you have to assemble, who can bring together sort of a very analytical framework, not only for data gathering but also for analysis and decision making, okay, on what compounds to move forward. So let's sort of step back a little bit. I think one of the things that you hear a lot of pharma companies do, and I think even biotechs over the past few years, is really starting to address early human dose projection in their discovery phases. And that is going to be very critical, because what it does for you is it starts to aggregate all the different data sets required. The most important thing is human dose. Okay. The lower the human dose, as low as you can get, the more safer the drug. And that is well known in the literature. I'm also one of the authors in one of the papers way back when that we published as well about low human dose and low uh, free drug exposures that really help us drive a much higher therapeutic margins. Very critical, especially in small molecules, right? Having analytics and having ways to address and understand human dose projections as you move through phases in discovery is critical. The more data you get, of course, the more firmed up this number is. But you can still start with a very nascent data set and understand whether a certain series has better propensity to get into the low human dose bucket versus another series. And then as you hone in more and more in the particular series, you're able to titrate more and more data. As you move from in vitro systems to in vivo systems, and then correlating the in vitro to in vivo metabolism, in vitro to in vivo PK and PD is critical. So the second part is translation, translation, translation. And that's where it's very hard, right? No translation system is completely foolproof. There might be a few there are a few predictive translational models. Some people are more lucky than the others that they can isolate cells from patients and ask functionally whether that's restored. That's what we try to do a lot within Rectify as well, for programs where we can get access to patient-derived cells and ask whether our positive functional modulators can restore back that function of the transporter is a very key event that de-risks for us what it means. And then how does it perform in vivo? And the translational models, both in vitro and in vivo, are critical. And then you have to aggregate quantitative pharmacology. That is the key part. I think that's where a lot of the decision making happens, right? Not all the data is going to be available to you to make the most important decision and judgment to move forward. So the collective experience of the team and what you've seen in the past, and does it address the need of the patient, and that patient population is critical. Number one. Number two, when you take that preclinical translation and go to the clinic, what is the earliest signal of efficacy you can get in the clinic? Can you be innovative enough to get a signal in a phase 1B MAD dose when you reach your last cohort? Can you get a signal of efficacy in a phase 2A POM over 2 weeks, over 4 weeks? And then how do you address dose selection in phase 2? What is the most important thing in phase 2 is to get the right dose. And it's hard, right? It's not easy to step in and get the right dose. So what data set will you have? What confidence do you have to move into a strong phase 2 POC that gives you an end of phase 2 meeting with the FDA? And you have a dose selected when you go to your pivotal trials. So I think that's important. Third is alignment with regulatory authorities. As you step in and you move in, that is going to be critical. In rare diseases, as you know, one of the challenges is recruiting, but also finding out in rare diseases is how strong the patient advocacy group is. What is the patient advocacy group? Where are the physicians? Where are the treaters? Who are the providers? And do we have the regulators in the same table for those rare diseases? Are they motivated, as the patients are, to allow us to get a drug moving forward for that rare disease? That is critical. I think PSC is ripe for innovation. It's a large, rare, unmet disease. It's devastating to patients. What people don't realize is that once you present yourself with PSC, the physician is going to say you're going to live under the fear of developing cholangiocarcinoma, a very rare bile duct disease. 400x risk when you have PSC to get that disease, to get that cancer. So lots of motivation to drive. So the question then with regulators is there a biomarker strategy for us to get to an accelerated approval, which is what we are working on right now at this point in time.
Wonderful. Thank you so much for that rundown. It's definitely a full basket of things, really a lot of considerations, and it really speaks to the immense amount of work that goes into drug development, especially in the earlier phases. So thank you so much for that. And in terms of I guess the current trends in the space of early drug development and also in liver diseases in general as well. So in those two kinds of arenas, what are things that you've seen that have evolved in the last couple of years and that are really helping or enabling innovations in early drug development?
I think the ability to sample and measure biochemical markers of liver health, liver injury is critical. Of bile duct health is also critical. So the nice thing about the liver is you can measure very quickly serum biomarkers and start to make assessment whether your drug is working early, right? But it is not enough because you got to start to make inroads into what are the key phenotypic measures of efficacy in that patient population beyond biochemical markers. What are the other markers of efficacy that you need, which the regulators might require for approval? What we are starting to see is, I think, a very strong approach to aggregate these markers of bile duct health and liver health to really paint a picture of early proof of mechanism in these patient populations across a number of hepatobiliary diseases, some better than the others. It's a spectrum of more firm data and some less firm, but that's something that's happening a lot. The other thing is imaging. Actually, it's funny that you ask, and have to roll out AI and imaging here. It is not really helping us in the drug discovery part of things in terms of identifying the drugs. But what's helping us is helping us understand when we get the data in humans how to map the biliary tree better. And it allows us to very quickly image whether our drug is actually working to improve bile flow and reduce biliary strictures in the biliary tree. It's a very complicated biliary tree, right? It's small ducts, it's large ducts, it's intrahepatic, it's extrahepatic. So having a very nice physiological view and a pathophysiological view that can be imaged to really show with drug and without drug, what the differences are is very critical. So all these imaging technologies are really helping us advance moving forward to developing drugs and getting the data in early clinical trials.
Wonderful. And a bit about your work as a venture partner at Atlas Venture. How does that work there inform and influence your leadership at Rectify Pharma?
Immensely, I would say. It's critical. Rectify Pharma was formed when I was a venture partner. I was on the board of the company. I was not the CEO. I was on the board and the SAB of the company. Helped form that company with by speaking to the founder of the company when that person was at MIT doing research on advancing assays for ABC transporters. Um, um, and that was only possible with the network and the support of Atlas Venture. Number two, Atlas Venture was a seed investor and helped sort of lead the Series A, along with Omega, Forbion, and participation from Longwood. So that helps. I think as a venture partner at Atlas Venture, one of my key uh, interests, passion, and strengths is venture creation. So, new venture creation, really understanding what it takes to translate early science and move it through, and how do you put a business plan together? What does that financing look like? And who is the talent you need very early on in the company to help? Remember, every company goes through multiple phases in its evolution. Early, mid, and late stage, right? So it's very important to have the right sort of talent in the company to help advance it to the next phase of development. Early discovery, one thing, preclinical translation, one thing, early clinical translation, and then, of course, late stage clinical development all require a different mix of talent and interests. And you've got to make sure that the interests align with where the company stage is at that point in time. So really, I would say venture creation and having the ability to draw upon the expertise and experience of other people, partners, venture partners within the Atlas network, which is a very large network, is critical. So number one, talent. Number two, able to really get your data firm, your ideas firmed and then vetted and understanding, is this the right plan? Because, is this the right financing plan? Do we have the right goals for the organization? Can we deliver X in a certain point of time? That's always a negotiation with the investors and the company, right? It's uh, the faster the better, but there's also a certain sense of realism that
Yeah,
needs to come in as well, right? And
absolutely.
stretch goals are very important for any company when you begin and through the mid
Yeah,
phases as well
indeed.
of company growth.
Yeah, and speaking about investors and stakeholders, are there any partnerships or collaborations that stand out or that are noteworthy that have really helped advance Rectify Pharma's work so far?
Um, Not yet. We've been really focused on incubating the. So let's sort of step back. In September 2021, we came out with a Series a large Series A, $100 million, to really build out the platform. Number one, addressing very quickly whether this platform is going to work to provide us small molecules that are drug-like that can restore protein function, right? And then it was a target space. But me being a drug development, drug hunter type, I I'm not someone who likes to spend a lot of time on platform building. Very quickly, sort of steered the company towards what does the product look like, what does the TPP look like? Is there value we can drive? Just not target product profile, but is there value in that target product profile? And is there a real unmet need? And number two, will people pay for this? Whether it's a pharma strategics, whether that's patients, whether that's providers, whether those are physician and physician scientists, what do they think about it? So having those conversations, which are not easy, right? You continue to have those conversations and you get the data and you inform your confidence on moving forward. That's going to be very critical. So those are the things that we've been doing quite a bit. And in terms of advancing our lead programs, we've done that on our own. Now I think is the right time for us to sort of think through, is there path for us for collaboration and partnerships with others? And we are in discussion with a few people right now.
Wonderful. And finally, I think these are like maybe two questions combined into one, but what is your vision for the future of early drug development in specifically in the liver disease space or in general as well? And how do you see Rectify Pharma leading in this space? And number two, what milestones can we expect from Rectify Pharma in the coming months and next couple of
Yeah.
years?
Oh, great. Maybe we can, if you have a half an hour more, I can give you my feel on what I think discovery and early
Yeah.
clinical development needs. But um, very quickly, I think it definitely needs experienced people. I think there's a lot of decisions that are made with not all the data on hand, right? You do the best experiments, you do the most information rich experiments. But at that time of making a decision to move forward with a candidate or not, with a program or not, with the indication or not, and with mortality or not, requires a tremendous sense of experience, which is what the board brings, which is what the investors bring, which is what the senior leadership on any company brings. Specifically to Rectify Pharma, I think what we are mostly excited about is our mechanism of action. This dual targeted ABCB4 BSEP small molecule has the ability to address multiple hepatobiliary diseases in a single pill. So the question really is, what is the stepwise strategy for us to de-risk this powerful mechanism in the clinic? And I think we are probably one of the few mechanisms that directly addresses the underlying cause of the disease, which is PSC, right? It's not saying we won't have efficacy in other diseases, hepatobiliary diseases that are well known, PFIC and PBC, but PSC is the one that really catches our attention. Very large unmet need, no approved therapeutic, and really presents a great opportunity for us to start asking that question. So I think for liver diseases, watch this space. I think we have a great mechanism. The data is borne out well. In November of last year, we presented in the AASLD meeting. We also had an R&D day that's published in our website. So definitely people can go and look at that, and the KOLs talking about this really unique mechanism. Looking forward to the most important thing for this company is transitioning into a clinical organization. So that's our goal this year, is to take our lead program and start our clinical trials.
Wonderful. And we will definitely be watching this space for Rectify Pharma, and really looking forward to seeing what's next for the company and what you'll be bringing to the space. Dr. Devraj, thank you so much for, number one, firstly, the work that you do and everything that you bring to Rectify Pharma, everything that you bring to this space of early drug development and liver diseases. So thank you so much for all that you do. And thank you for your time to share everything that you do with us. We really appreciate it.
No, thanks a lot, Ayesha, and thanks for Xtalks for giving the opportunity for me to discuss what we're doing at Rectify Pharma and this environment of biotech,
Yeah,
entrepreneurship, and drug making new medicines. That's we need it,
we need it.
and it'll
Yeah.
continue to go on. So
Absolutely. We need it. It's exciting, and ultimately keeping the patients in mind there, really doing life-changing work. So once again, thank you so much.
thanks a lot. Take care.
You too.
See you. Bye.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.